Hello and welcome to my training video for TAS2R38 genotyping analysis. This is one receptor with its own little video for analysis, which may seem like a lot, but in fact, the methods for this type of genotyping are a little abnormal to your day-to-day SNP analysis, and if none of that's really resonating with you, that's totally okay, because I'm about to go through everything, so you're in the right place. So let's get into a little overview. As I mentioned, we'll be talking pretty extensively about TAS2R38 and its genotyping today. I'm going to give you some background information on the bitter taste receptor itself and why it's a primary focus point of the read lab, and then after that, we'll be looking at the genotyping files that get spit out of the machines. Listed here, you're going to see the two RT-PCR machines, which stands for real-time PCR, which refers to the group of machines that can not only amplify the DNA, but also make readings in real time about what's happening with the DNA. So the first machine here is the Step 1 Plus, and the second one is the QuantStudio 12K Flex. So the 12K Flex is a little bit bigger and can do more experiments with different plate sizes, and the Step 1 Plus is more limited to 96 well plate experiments, and it can't run things like microchips, for example. So the Step 1 is a little bit older, and it's less exciting to look at, but it's an old reliable in our lab, so we still use it. We'll investigate the files that come out of those softwares, and we'll go through the process of exporting and what the raw files look like when they're on the machine software and afterwards. After we do that, we're going to examine the main methods of data analysis. So there's one that is sort of through our studio and it's more automated, which is what I prefer. It's a code approach and focuses on making sure that it's an efficient and correct process, as opposed to some of the mistakes you can make when you're manually coding the data. So you can always, if you have problems running the code or it just doesn't make sense to you, use a manual or Excel forward method of analysis. I personally think that it is more irritating than R, and that's actually pretty difficult to do, to be more irritating than R. So I definitely would recommend at least trying the R code and seeing if it works for you. So let's start by introducing TAS2R38, which is a bitter receptor. When we're talking about this in this context, we're talking about the gene rather than the actual receptor that encodes for TAS2R38. And more specifically, we're focusing within a particular exon or coding region of that gene. So in this exon, we can see there are 3 points labeled. We're going to call them single nucleotide polymorphisms, or SNPs for short. And these are titled with numbers that begin with the letters RS. And so that's how SNPs are identified. It's RS followed by a long string of serial numbers. And so these specific ones that you can see in the diagram are integral to how certain chemicals bind to the receptor in question. So two chemicals that we use in bitter taste testing pretty commonly, which is why TAS2R38 is really important, is PTC and PROP. So you might recognize the word PTC in its case strip form that you might have done in a class in high school or college to demonstrate that some people in your class have different phenotypes of taste, and comparing that to your genotypes if you were able to get them genotyped. So some people cannot taste PTC at all, and some people taste it, but it's sort of middlingly bitter, and some people find it extremely bitter. So that's why this receptor is really nice to look at, because we have that nice sort of spectrum of where people can fall phenotypically, and then we can actually see that parallel in the genotypes. So as I said, we use these 3 points of mutation, the SNPs, to identify the genotypes behind the phenotypes of bitter tasting of PTC and PROP. Each SNP causes a change to the triplet code that tells the cell which amino acid comes next in the polypeptide strand that it's making. So the end product to be folded into the protein TAS2R38 is now different depending on which alleles you have at these 3 SNPs. So also remember how I said that the SNPs are really important for how TAS2R38 binds to chemicals. So the reason that it is important is because each of these amino acids changes the binding site area specifically. So everyone has a specific recipe for each protein from each one of their parents. So you have 2 different recipes for types of functionality in your proteins. For me, I have 2 different sets of alleles for my mom and my dad. One is a really good fit for molecules like PTC and PROP, meaning my brain gets a super bitter reading from some proteins, and the other protein is not as good of a fit for these molecules, so it's a softer bitter read. I can't tell which one comes from which parents from looking at my overall genotype, but my prediction is that my dad has the variant that binds less well due to his love of many bitter foods that my mother and I are less interested in. So you'll notice on the previous slide, I had this letter combination. So this whole string is called a diplotype, and the slash divides it into 2 haplotypes. These letters are not all nucleotide letters, so you can see they're not A, T, C, and Gs. So we aren't actually referring to the letter at the SNP spot in the DNA code, but we're actually referring to the resulting amino acid in the polypeptide that would be changed depending on what allele you have. So all these different letters are the abbreviation of the amino acids at the 3 SNP sites, which is why it's the 3 and then the 3 on either side. So the haplotypes come from each one of your parents, but since we're diploid organisms, we have 2 copies of each chromosome, each gene, each receptor recipe. So we refer to the composite of your 2 parental recipes as your diplotype. So what we can infer from looking at a diplotype is that the 2 most common haplotype forms are actually going to be PAV and AVI. So when we look at the diplotypes, we're going to see usually a combination of those 2. So that tells us directly whether somebody is a het, a super taster, or a non-taster. But even though most people are homozygous or heterozygous of these 2 haplotypes, there is so much diversity in genetics, but especially things like taste. So as you can see from my diplotype in particular, there's actually much more complexity than AVI and PAV. So remember, the letters in the haplotypes stand for amino acid results from the SNP changes, and the A and the V here stand for alanine and valine. I could keep going, but it's easy to Google the letter codes for amino acids, and if you're really interested in that, you can do that. So as I said, these amino acid changes are causing effects in the binding domain. All amino acids have slightly different structures and chemistry. So if you sub one in for another, it can completely change how a molecule can bind to the receptor. I like to use the metaphor of different chair shapes. So if we say that the PTC and the PROP molecules fit super well into the super taster chair, they also fit okay into the person who has a scramble of some functional and some non-functional alleles, but they can't comfortably fit into the non-taster chair. Before we go forward, I'm going to briefly touch on Hardy-Weinberg equilibrium. So linked right here is a Khan Academy lesson that you should definitely watch as a refresher on this concept. Most people don't do Hardy-Weinberg enough to really have the pulse on it, but since I'm going to take a much more glossed over look at it in this PowerPoint, you should definitely give it a look over, watch the Khan Academy lesson again, and refresh yourself. At the end, we're going to use an HWE online calculator because it's much more efficient and it doesn't leave any room for your sort of human statistical calculation error. Basically, most important things to know about this type of calculator is you're going to need the head count of the different breakdowns of genotypes, the common homozygotes, the rare homozygotes, and the heterozygotes. And then we have to do this on a SNP level specifically, not the haplotype level. So for TAS2R38, you're going to repeat the process 3 times, 1 for each SNP. So we can see here there's a little cheat sheet for which homozygotes are common versus rare, and that should help you do that count, the head count. So if you need to look back at this later, you can. So the reason we have to do it on a SNP level as opposed to just looking at AVI haplos are rare and PAV haplos are common and doing the Hardy-Weinberg equilibrium that way is because, again, there's so much diversity that you aren't capturing with that, because a lot of people out there have some rarer haplotype, like myself. So Hardy-Weinberg is intended to be used on that allelic level, so you have to do it at the SNP level and repeat it 3 times. So moving forward, we're going to look at the 2 machine softwares and how to look at the data on there, but also how to export it, which isn't particularly difficult, but just so that you have that visual of where the export button is and all of that. So it's really important to note that the allelic distribution plots that are shown in said software are not always perfect. Sometimes not all the combination of homozygotes and heterozygotes are present, and if they're not all present, then the machine can get very confused about where is what group and all of that. So it can make some really wrong calls. So you have to be alert to that. And we'll talk about, when we're looking at the actual software, what that might look like and how to cope with that. So the different softwares are pretty similar. It's not a huge learning curve between the two. What I want to show you is the file when it comes up on the software, and then how to download it and what the file looks like when you open it in Excel and haven't touched it yet. So the first thing you'll do is check the control samples, because you do not want to go any further in the analysis if the controls look messed up. So again, here's a cheat sheet of controls recommended to use and what to look for in the unanalyzed data. So we're going to pop open the Quant Studio 12K Flex software, and you have to open from within old files. So I'm going to be going in here and trying to desperately find the file that I'm going to open. And it takes me a few moments because I don't know why, but I could not find the folder I was looking for. Okay, there we go. I'm opening my sample files, and I'm opening the specific one for SNP genotyping. So as we wait here, there we go. So the file comes up. You can see the first thing is the plate map of the different samples, like where they are on the plate. I don't usually fill up a whole plate because I'm running all the 3 SNPs, but I sort of repeat them across the plate and then run the different SNP assay in that different part of the plate. So you can click on the drop downs at the top left to get to the different SNPs and look at the different allele distributions. TAS2R38 is usually pretty clean, so it's usually not very, very difficult to sort of see if something is desperately wrong. If it doesn't look like a clean 3-part distribution plot, then there's maybe something a bit off. Either you have a weird population that you're looking at, or something else. So here we're going in to click the export button. We're showing where we want to save it, and then we're going to edit the title of the file there in the top right to change it to what we want it to be. And then we're typing it out. And then on the far top right, you can see there's like a little drop down that says XLXX. So you want to select the Excel workbook drop down there. So we're going to click Start Export. Then I'm going to open a file. That was the wrong one. I had that open earlier. I'm going in. I'm going to go find the file I just downloaded. And there she is. Come on, you can do it. Okay, so this is what the file looks like when you just download it. It has this whole top section that tells you about the settings. We don't actually need that for the analysis. So the first thing I do is I delete that, and then I save it as my new editing file. So yep, I'm going to change its name, and I'm saving it. And then you'll notice that there's no sample listing on here, so we don't technically know which samples there are, but you should always make a plate map Excel file, which I've made a pretend one here. So I'm just going to copy and paste that in. All right, so now we're gonna look at some data that is less nice looking. Okay, so you can see here that I am in an open array file actually, so it's got 64 different SNP assays that it ran, but a lot of these SNPs are just not as clean. They just don't come out as well. So there's all these like weird distributions, and you just really can't tell necessarily who's in what group. To my eyes, it looks like the left side sort of linear group is probably at least mostly homozygotes in the allele 2 group, but that middle group is being called as allele 1 homozygotes, even though it's likely heterozygotes and there are no allele 1 homozygotes. But again, this is just to emphasize that the computer is not always perfect at calling things. It doesn't always understand the complexity of a cohort that we could be looking at, where, again, like one of the groups is just completely missing. Here we can see a data set in which there's basically just one group of genotypes. So this is probably all kind of one group of people. Again, it's not super clean, but this is just the nature of the microchip. So the most likely thing is that these are heterozygotes, as opposed to them being all one of the homozygotes, especially looking at the way that it's distributed. So yeah, after you make these kinds of edits, you're going to want to save before you export and save the file, probably under a new name. Okay, so we're going to be jumping into looking at the other software for the Step 1 Plus. This is the Step 1 software, and it's always going to give you an error message when you're on your PC in the office that you're not connected to the machine, and you just tell it that that's okay, and you click the continue button, which I'm taking a long time to click here. There we go. Okay, so this is what it looks like on like its home screen essentially, and you have to do the same thing where you open the files from within the software. So you open the software first, and then you go and you open the files. So there I am going to find my file, which again will probably take some doing because clicking through the many shared drives and Google Drives that I have takes me time. Okay, so it looks like I found it. This is my example file that's in this genotyping analysis file. And okay, so I'm looking at another TAS2R38 experiment, but this one was run on the Step 1 Plus. So if I click off of that, yeah, it'll unselect it so I can actually see the different groups. So this one's like looks a little less clean, but it's also got less people in it, so that usually makes for less tight clustering. But it also could be the difference in the machinery and whether or not it had been recently calibrated, so they might just like not be as tight or clean as we might like. Again, this machine is older and it does actually get a lot of use, so I may not have calibrated it recently, especially for the VIC-FAM dyes that we use in SNP genotyping. Okay, so you're gonna go to the left side and click the export button, and then you're going to want to do the where am I going to save it search. So that's what I'm doing right now, is I'm picking the correct location in a folder that I wanted to put the file into, and I already renamed the file to what I want it to say. A lot of the time it'll do a default name that's not very descriptive. So there, and then we click start export again. Now that we have talked about data exporting, we're ready to begin with data processing or analysis. The first method we're going to look at is the one I mentioned at the top of the presentation that uses R code. As you will notice, we have already done step 1 here of deleting those top rows off of the sheet. So we've also performed the second step of importing a plate map that we should have typed up before running the experiments. Remember, when we copy and paste in that plate map, we really need to make sure that the right samples are getting pasted into the right wells. So in the process next, we're going to open our R project from the folder R analysis and copy and paste our plate mapped export file into this R analysis folder so that we can access it when we're inside RStudio. When we run the R code, we're going to end with exporting what is called a wide file. And this is just the data arranged in a way that has all the SNPs and amino acid calls in a row next to their corresponding sample name. So they're all together and grouped, as opposed to all the calls being in a long list next to their sample name and then their SNP name. So you'll see this when we run the code and how it gets grouped together. Okay, so I am going to open the R analysis folder. And from here, I'm going to go into TAS2R38 genotyping. I'm going to open up my project here, but I'm just going to note that this is our file that we exported earlier in the video. So I copy and pasted that in here previously. Okay, so I'm going to open up cleaning and calling TAS2R38.R, which is a script. Here we can see said script. And the only thing I'm going to go through and change is make sure that this file that we're putting in at the beginning is correct. And we're also going to import the file because I'm going to pretend that I haven't put this file in the system before. So if you had just copy and pasted in your file, now you're going to import it. Now it exists in our directory up here. And I'm going to make sure this is the right file. Yes, that should be correct. So this next line that I'm running is letting us know that the key table that I'm going to be using, which will tell us which SNPs and amino acids are being called, is staying the same. I'm just going to show you it because it's easier to see. Here we go. Yep. So this is it. It's a little baby table. It just tells us RS102 is C and T for allele 1 and allele 2. And RS172 is AG. And RS713 is CG. Okay, so that's our key that it's referencing, and it'll reference this later to make allele calls. We also need to first run this, and that's basically libraries are just telling the code that it's going to need to reach into the ether into these specific groups of tools, and it's going to use tools from these in this code. So that's all we're doing. And I didn't need to run that before this, but normally I just run from the top down. So the next thing that we're going to do is to clean up the raw file that we just input. And we're going to delete a bunch of columns that we don't need. So now if we look at the del call, which is what I've just labeled the raw file with all these things deleted as, we can see that I've cleaned it up. It's now just the positions, the samples, the SNPs, and their calls, because we don't need all of the other information right now. So the second thing we're going to do to clean up a file is we're going to rename a bunch of the columns. So this is how you rename them. I'm going to click enter. And now if we look at clean, you won't notice a huge difference, but all these things are formatted in a specific way. Now we're going to correct a title name in the key reference table, and I could have just done it the right way when I made the table, but I didn't, so I just put it into the code to clean it as well. So now we're going to join the table that we made and cleaned by the key. And when we click on allele merge, which is what you called it, now we can see that it recognizes that ASCII is something that it has in its table, and every time that SNP occurs, it tells us what's allele 1 and what's allele 2. Okay, and we're going to need this later. It doesn't actually make a call yet, right? It's just giving us the right framework. So this is the function that we're going to use. So now that we've put those two columns in that tell the computer what the answers, quote unquote, are, we're going to run this function that's going to generate a call for, if it says homozygous allele 1, then it's going to print something else, okay? So it's going to print allele 1, allele 1, which is already in that other column. So we're then going to add basically a column at the end that is this call from this function. It may look a bit daunting, so if you don't quite understand what it's saying, honestly, me neither, but it does what we need it to do. So now we're generating an empty sort of data table out in the ether that is the size of the original data frame, and we're making a new row for it. So now we're going to loop through that data frame and then make those calls that we told it to make with the function. And then the final part here is we're going to bind the allele call that we made to the original allele merge table. Okay, so let's look at final data because a lot has happened. Okay, cool. So even though we've run a lot of lines of code, all we did was generate this allele symbol, which just is the call for every single one. So you didn't have to do that by hand. Hooray. At this point, I just like to write the CSV for just the SNPs, and just keep the file here. Every time you use the write CSV function and you're using a different file other than my example file that I have input here, you'll just change this name as well, just like you did at the top when you were inserting the raw file input. Now we're missing the amino acid calls for the haplotypes and the diplotypes. So I'm gonna hit enter on this line and run that we're telling it. When I say amino key, I mean this specific table that's in our directory already because this is a previously used project. Now I'm going to run this line, which is joining the amino table with our final data. So we'll look at it. Amino merge. Hooray. Again, it's saying that these things mean these things, and so it's going to duplicate that column, and we'll clean that up later, but we don't have to do it right now. Final amino clean is me deleting those columns, and then I'm writing that CSV. So as I said at the beginning, I'm going to make a wide file. So I need these new libraries to do this part of the code. I keep the code kind of separated out so that you don't have to do all of those different things if you don't want to, and you can get those CSV spit outs at each stopping point. So our input is going to be, let's scroll back up, right? So this was our final file that we just spit out and wrote a CSV of. So now that's our input. When we run this line, we're saying we're removing the duplicates from it. So that's why I called it rem duplicates. And the function is unique. And then we're telling it which thing it's uniquing. And when it uniques, it's just deleting every duplicate. So I'm going to run ydata1, which is our remove duplicates file. And I'm going to say pivot wider. And ID calls means what are we using as the baseline for pivoting wide? And what are we using for the new column that we want to be sorted by sample name? So the SID, we want to be the new column, and then we want the values in those columns to be the allele symbols. So we'll look at it really quick just so you know what I just said. Okay, so we have the sample name here, and then all the sample names we've grouped their SNPs together so that you can just look across and see all the different SNPs. Perfect. Okay, ydata2 is going to be a similar theme, but instead we're grouping by amino symbol. So we're grouping by the amino acids this time. So we've made two separate tables. One of them looks like this, and one of them looks like this. And I like to close out of my tabs every single time because I don't like tabs. And now we're just going to join those two tables together by sample name so that it groups all the samples together. So we're going to hit enter. Alrighty. And then write CSV again. And I'm going to open that in the actual CSV form. So I always save my project. Save and everything. And I'll X out of here. Save again. Save, save, save. And then, yeah, here's our thing that we downloaded last. This is our last guy. Okay, cool. So you can see here, or you're going to see it'll tell you like, oh, this came from the one data set and this came from the other data set. So they're calling them X and Y. But I would just change these to be like 713, 598 amino acid. Or whatever. But you change your table to see how you fit. Now, all of the data is aligned properly, and this allows us to very easily make the diplotype calls. So what I'm going to do is I'm going to sort by column. My data has headers. I'm going to sort by this, and then this, and then this. And you can sort by those ones or by the amino acids. It doesn't matter because they are directly congruent. Okay. So now we've made it easy for ourselves. We can see that it's grouped all the patterns kind of together. And so we can see that this person is ABI, ABI. This person is also that. And then what I like to do is just for those groupings, I just click and drag down. And now we see an AVI PAV. And you're just going to go down the line as such until you meet your next group, which is PAV PAV. I'm just going to save this and close out because I'm going to save as because I just realized that edits, and we want to save it as a workbook. There we go. And now you can freely edit and do your analysis and have this final nice table. Now we're going to cover manual analysis. This kind of analysis may make more sense to you if you're unfamiliar with R and coding as a whole, but in practice, it gives you a lot more room to make human mistakes that you may not even notice. So when you're doing manual analysis, you have to be really, really attentive and triple check your plate map and everything that you end up doing and making changes to with the Excel document. I'm going to insert resources in this PowerPoint, but you are just going to follow the TAS2R38 genotyping analysis Word document, which is a written manual for doing this analysis by hand in Excel. You may also use the TAS2R38 key spreadsheet for making sure you have the right calls for alleles and amino acids. Again, you should reference these tools. The common versus rare sheet will help you when doing Hardy-Weinberg equilibrium, and the diplotype table at the top will help you see if any of your rare diplotypes seem to be overly represented in your cohort. So now we're going to move forward to the HWE equations. They're listed here, and on the next page we'll delve a little bit deeper into what the P's and the Q's are. If you're still confused after my brief explanation, please look back at the Khan Academy video or find online resources because, again, I'm not going to cover this super in depth. So this is a quick example I made. It's fake of 18 individuals being genotyped for RS713-598. You can see that we are going to count the presence of each allele by multiplying homozygotes by 2 and counting heterozygotes as 1. Then we total that for each, total the 2 allele counts, and calculate the percentage of that total for each allele, which then become the P and the Q. We calculate the P squared, 2PQ, and Q squared, and then we multiply those fractions by the total number of individuals to get our expected counts of each group. So now we'll take the expected numbers and we look back at how many homozygotes and heterozygotes were actually seen, and we'll calculate the chi-square to see if this distribution is adhering to normalcy. Okay, so I went over how you would hypothetically do it by hand or by Excel formulas, but I don't believe in doing that kind of work when there are so many more efficient resources that are likely better at math than you or I are. I listed the head counts again, so we can easily reference them. So we're going to open up this online calculator and let it do the work for us so that we can just see the results and download them. Okay, so here we can see that we're on the website. I'm going to enter in my common homozygotes, my heterozygotes, and my rare homozygote counts. Okay, so if I click calculate, I can scroll down and I can see a bunch of information being spat out. And I can see more information about Hardy-Weinberg equilibrium. If I scroll down, it'll give examples, etc. So you can follow along if you need more help and resources with that. It's also going to calculate my statistical analysis for this, so the chi-squared number. And if I want to save this for later, I'm going to click the PDF button and then click download as PDF right there. So it's going to open in a new page. And now I've downloaded this PDF version of my statistical analysis for Hardy-Weinberg. So you can close out of that. I recommend using this approach and not doing it by hand. So let's proceed. Now we've analyzed the different SNP calls and we ran HWE. So we need to do a head count for the diplotype results and compare that. If you aren't sure how to call diplotypes and you followed the R coding approach, pop open the TAS2R38 genotyping analysis doc and scroll down to number 16 in that manual. So you will get detailed instructions there. Once you call the diplotypes, you're going to calculate the percentage of each type in the groups and compare that to this chart. Or there's one embedded in the genotyping analysis doc. That's the end of our delve into TAS2R38 genotyping analysis. Hopefully this more visual approach to it was helpful and you feel more ready to get started. So the next video is going to be on some more different SNP genotyping analysis points and things that I would like you to know.